The second part of this chapter is talking about sterility in general and specifically on male sterility. Male sterility means the failure of sterility in general means that the plant is not producing a fertile pollen or anther. And it is different than incompatibility, that we have a normal pollen and anther and ovule, but it is not setting seeds. While here, no, there is production of non-fertile pollens or eggs. If the plant produces non-fertile pollens, we call it male sterile. If it is producing non-fertile egg, we call it female sterility. But female sterility is not stable and it is not utilized in plant breeding. Therefore, because we don't have female sterile, we have only male sterile plants, we always use the male sterile flowers as female flowers. Male sterility can be caused by two types or by two means. First of all, it can be directly affected or controlled by specific genes, and in this case we call it genetic male sterility. Or sometimes it is indirectly regulating by gene action, where directly the factors are found in the cytoplasm, and we call it cytoplasmic male sterility. So we have the genetic male sterility, which is controlled by certain genes, and cytoplasmic male sterility, which came from cytoplasmic influence, but it is originally under the control of regular genes. Genetic male sterility. As I mentioned, it's controlled by genes that inhibit the normal development of anthers and pollens. We don't know exactly where is the inhibition happened, because we have different genes and the different plant species differ in the inhibition stage. In order to utilize any male sterile genes, it should be very effective and it should not produce any viable seed. Therefore, it has to be complete sterile with or very, very, very low percentage. And the problem is that this effectiveness is affected by environment. Therefore, any gene that has been utilized in plant breeding should inhibit all seed production. In addition to that, it should be stable in a wide range of environments. In other words, it should inhibit 100% seed production in any environmental conditions. Male sterile genes are producing male sterility when they are in the recessive case. Therefore, capital male sterile, capital MS, or capital MS, small ms produce male fertile plant, which means that we have dominancy. The only case where we produce male sterile plants is when we have two pair of recessive alleles. Therefore, according to what we have here, we cannot produce pure population of genetic male sterility because it will not regenerate itself. Therefore, we always keeping the population in the case of heterozygous, where in the next generation it produce both male sterile and male fertile plants. Male sterile genes have been identified in many species. For example, rice, wheat, corn, barley, and it is found spontaneously in the field, while we can induce it using a mutation. When we look to the anthers of the male fertile plants and the male sterile plants, we can recognize them very well because we couldn't find the anthers, or sometimes it is small and rudimentary one, while the male fertile anthers are having a normal anther size. In plants in general, we could recognize till now about 25 non-allelic male sterile genes that found in nature. As I said in the previous slides, we cannot maintain pure sterile plant population. We want to see how we can maintain sterile population. Let us say that we are crossing male sterile plant with a male fertile plant. In the F1, we produce heterozygous plant, which is male fertile plant. Then if we self it, because we are talking about self-pollinated crops, if we self it, we'll get 25% male sterile and 75% male fertile. Out of this, we will have 25% M capital, 50% M capital, M small, heterozygous. Now, when we self it to the F3, we will find that the ratio will be 33% MS capital, MS small, and 60% heterozygous MS capital, MS small. And this ratio is coming because the only source of pollen is coming from the male fertile plant, which is 70%. And if we calculate the allele ratio, we can find that the MS capital to the MS small will be 33% to 66%. So the pollen source genotypes will determine the next generation ratio. So then we self in the F3 to the F4, and in this case the male gamete will produce 50% MS capital and 50% MS small, which end up with 50% MS small, and 50% MS capital, MS small, and this ratio will continue till the end any generation, maybe after 100 generations. So we repeat it from F4 to F3, and we found that it is producing the same ratio. So we will keep this population, which has 50% male sterile plants and 50% male fertile plants, and each time we harvest the seeds from the male sterile plants only. And in this case, we are sure that we get 50% male sterile and 50% male fertile, and in this way we can keep the male sterility in the population. Sterility was very important for us as plant breeders, and we can use it in many ways. First of all, I don't need to use any emasculation when I do hybridization, because emasculation of self-pollinated crops need a lot of time, need a lot of labor, and not all the people can do it correctly. So it is really very difficult process, so we don't need it. And male sterility can be induced in many varieties through back cross. And if these varieties are involved in crossing, then we can use these varieties or the male sterile plants as female plants, and in this case we do use hybrids from the male sterile without using or utilizing without doing any. The second way we can use male sterility in plant breeding, that sometimes we need to increase natural cross-pollination in self-pollinated crops because we need more recombination. So this method provides a means to increase cross-pollination in these type of crops without using hand pollination, which always limited the recombination process, because we need a lot of crosses to do pollination, and the season is very limited in time, and we have to do a lot of crosses in very short times. And this has been happened in two ways. First of all, we can produce large number of strains. Some of them are male sterile, some of them are male fertile, and we let them cross with each other, and at the end we can harvest all the seeds and bulk it and plant it next time. Or we can use the male sterility to facilitate recurrent selection, and when we reach recurrent selection, I will explain for you what recurrent selection, which means that we are doing selection each time. After we develop the original crosses, or what we call population, the good male sterile plants are selected from the segregated population, and the good male fertile plants are selected. We plant them, we let only the selected plants cross to each other, and we produce a new generation of heterozygous plants that can be selected each time. We select male fertile and male sterile and cross them with each other freely without doing any hand emasculation, would take time and efforts. The third type of utilizing male sterility or male sterile plants is to facilitate commercial hybrid seed production. As I said, usually we don't need any emasculation in these male sterile plants, and we need to produce high number of seeds, so we can incorporate the male sterility in one of our parents, and leave the other as male fertile, and let them cross, and the F1 produce, or the hybrid plant produce, we can plant it in the field. Theoretically, this point is excellent, but there is a lot of difficulty when we want to maintain a pure male sterile line, and it has been utilized in corn, and it is not easy to maintain the pure line, and that's why the male sterility has been not utilized widely in a lot of plant breeding procedures. By this, we are ending the male sterility, and we will discuss in the other part, in the last part, the cytoplasmic male sterility and the other related subjects.